All right, so it's my pleasure to talk a little bit about the things we're doing in our lab, which can be broadly named computational microscopy of the cell, with the overarching idea that what I cannot simulate, I cannot understand. And here you see in this movie a zoom of the cytoplasm of a cell, where with this computational microscope, we can really look in detail at the motions of all the molecules that constitute the cell and that give rise to the functions of a cell. So here you see a bunch of proteins and metabolites and some of the genetic material, this yellow DNA strand. So this is what we are doing, and we're both doing or looking at applications, but also we spend a lot of effort in trying to build this computational microscope to make it as accurate as possible. So I will talk a little bit about that and also then give some examples of recent application areas that we are working on. So let's see if we can get this to move to the next slide. That does not seem to be working, so. Seems like the screen is frozen. Let me see if I can go back to the normal pointer, because I get this message that I need to get rid of. Okay. So yeah, the computational microscope can operate, of course, at different levels of resolution. Some of you are familiar with doing quantum mechanical calculations, whereas others might work on the continuum scale. But as you will see in this list, you need kind of multiple techniques to bridge really from the scale where you can describe chemical reactions occurring, which are, of course, also of central importance to the cell, all the way to continuum models if you want to study the cell as a whole and maybe cell-cell interactions. But we are mostly interested in this intermediate regime where we can still follow the individual molecules diffusing around and interacting with each other. But we use this as we do this at a coarse-grained level, where we simplify the description of our molecules a little bit so that we can really go to system sizes that are up to micrometer length scales, and where we can study time scales that are up to millisecond kind of regimes. Now, for those of you who are not familiar with the underlying technique of molecular dynamics, here is just one slide showing what basically molecular dynamics is about. It's very easy in the sense that we're solving Newton's equations of motion for a number of interacting particles, which can be the atoms that you want to study, or if you coarse-grain your system, as we do, these are groups of atoms or coarse-grained beads. So they interact with each other, and then with Newton, you can calculate the forces between the particles, and this leads to particles undergoing accelerations. So if you start with a certain configuration, as is depicted here, then you can propagate that as a function of time. So you calculate the forces, the accelerations, you get a small displacement of all your particles over a certain time frame, a small time interval. And you can do that then many time steps in a row, and that way you generate a trajectory of your system, and you can then visualize this nicely, and this gives you then the movies that I already showed you. Right. So of course there are some challenges to run this realistically. So first you need to describe these forces between the particles accurately, and for this you need accurate force fields. The second challenge is actually to generate enough of these configurations that you have that you can actually calculate equilibrium properties of the system that you are actually simulating. So this is the sampling of the phase space that is available to your system. Now, to do this, there is traditional people use all atom force fields like CHARMM or AMBER or GROMOS. You probably heard of these kind of force fields. But in our group, we have developed these kind of coarse-grained models, and the force field that we're using in particular is called the Martini force field, that I will explain also in some more detail. But the principle of coarse graining is easy, as shown in this slide. So instead of representing all the atoms in the system that you are simulating, as is here shown for a simple double-stranded DNA fragment, you replace groups of atoms with beads, coarse-grained beads. And so you have a change in resolution, so you have less particles that you have to compute, which makes it, of course, then much faster. And that is the main reason why you want to do this kind of coarse graining. So the Martini model that we developed considers on average about 3 to 4 heavy atoms together with the associated hydrogens as a single coarse-grained bead, as a single interaction center. And thus, we have created a very efficient model for doing these kind of coarse-grain and MD simulations. The underlying idea of the Martini force field is the use of what we call a building block principle. That means that we consider fragments of a certain chemical property or a chemical nature. We consider them as our beads, and irrespective of to which molecule these kind of fragments belong. So in this example, we have a fragment that consists of a number of methylene groups, and they are represented by a single coarse-grained bead in our Martini model. And this is the same bead, irrespective of whether this fragment is part of an alkane or a lipid or an amino acid or whatever molecule you want to simulate. Of course, you need many of these type of beads because you have many chemical fragments that you would like to represent. And so we have a family of these beads that range from apolar bead types to intermediately polar to polar, and also some bead types that are representing charged groups. They come also in many subflavors. I don't have the time here to go in all the details of this Martini model. But what we then do is, if we want to simulate, in this case, a lipid molecule, we can place these beads to the fragments that we want to represent. So we have, for instance, the very apolar beads, the C1 beads. Those are the most apolar beads we have in our Martini family, are used for the most hydrophobic parts, the lipid tails in this case. And then we have, say, the charged group of a lipid is represented by one of these Q particles and has also, in this case, a positive charge for the choline group and a negative charge for the phosphate group. And then we have the glycerol linkage region, which is of intermediate polarity. So this idea is stolen from playing with Lego, which I did as a kid, many hours, where you have a number of selected Lego bricks that you can then unite to a large variety of different complexes. So, of course, the essence of the method is that you calibrate the interactions between these different bead types, and for that we rely on experimental thermodynamic data. And there's a lot of data available that tell you how certain small chemical molecules or fragments, say, partition between different kind of environments, between polar and apolar environments for all sorts of different solvents. And we use that data to calibrate the forces between these different particle types. Now, we also need bonded potentials. Bonded potentials are used to bind fragments together that are chemically bonded, because in a classical approach, we don't treat the electrons as such, so we need to capture the bonds by simple harmonic type of functions. And so also at the coarse grain level, we do that. But now these bonded interactions typically are parameterized to match conformations of the same molecule, but then sampled at a higher resolution, say with all atom simulations. So you can do that for small systems, and you get a feeling for the conformational freedom of your molecule, and you can capture that with effective bonded potentials. Now, then there's a lot of testing. So in this case of a lipid, you can see if you can put these lipids together with solvent beads, if it actually self-assembles into a lipid bilayer, which it is supposed to be doing. And you can see that depicted here, that you can simulate this process. And then you can also calculate all sorts of properties of these bilayers and see how that matches to experimentally known properties like the thickness or the area of the lipid. So we've done this extensively. Originally, Martini was parameterized for lipid systems and only later extended to other type of biomolecules. So this has been an iterative process, but eventually we managed to get this model working and capturing the properties of lipid systems quite accurately. As I said, later the Martini force field was extended to be able to represent all types of biomolecules and also other kind of molecules that are interested in more material science applications like polymers, nanoparticles, etc. So also water actually is represented, and using the same 4-to-1 mapping strategy. So 4 water molecules are represented by a single coarse-grained interaction site. We also have a kind of a more advanced Martini water model where you also add some charge beads, so you can capture the orientational polarizability of water, in particular for applications where electrostatics are important. So the key features are summarized here. So we have a chemical specificity. Martini still has a level of resolution where you can really distinguish between all the different amino acids, between different type of lipid tails, whether they're saturated or unsaturated, etc. It is much faster compared to all atom models, about 3 orders of speed up, and this is the main advantage. And the way we parameterize the Martini model using this building block approach makes that it is a very compatible and versatile model, and also pragmatic in its use, and people can easily add different type of molecules and parameterize these models, and then automatically they will be compatible with all the other molecules that are already part of this Martini dome. So the parameterization, again, is a kind of a combination of a top-down approach where you use thermodynamic data mostly to calibrate the bonded or the non-bonded interactions, and then a bottom-up approach where you rely on more detailed atomistic simulations to calibrate your bonded potentials. All right, one slide just to illustrate the progress that we've been able to make over the years, partially because we have been introducing this coarse graining, which really allows you to study much larger system sizes and timescales, but also, of course, due to the increase in computer power over the years, as well as improvements in the molecular dynamics software. So when I was a PhD student in the 90s, I was already simulating lipid membranes, small patches of just 50 lipids or so with an all-atom model, struggling to reproduce properties of lipid membranes at the time. But then over the decades, we started to have better force fields. We could embed membrane proteins. And now we're at a time where we can really study and simulate the interaction between many different proteins and many different lipids in realistic models of, say, a cell membrane, which is still one of the core application areas of my group. And as I will also show in this talk, we're now approaching the area where we can really simulate at the whole cell level. Okay, so overview of the rest of the talk. I will give 3 application areas, examples of applications that we've done, simulating ligand binding to the Photosystem II complex, then modeling of gene transfer, and then finally, as promised, a perspective on how we reach the whole cell scale. Okay, the first application is already a bit older one, but I think it's still a very nice example of what can be done with brute force MD simulations using this kind of coarse-grained models. So here we're looking at the thylakoid membrane, and particularly what happens to the Photosystem II complex, one of the key protein complexes that are found in this thylakoid membrane. And yeah, we are going to follow the dynamics of, in particular, a small ligand, which I will introduce in a minute. So where do we find these thylakoid membranes? Well, they're part of the chloroplasts. So chloroplasts are kind of the light harvesting organelles that you find in any cell. And also in certain bacteria, they have similar kind of organelles. So this is a small organelle that has these internal membranes, including these thylakoid membranes. And if you look in more detail at these thylakoid membranes, you see that they are packed with membrane proteins that perform all sorts of enzymatic activity. And it starts actually with the Photosystem II complex, which will also be the topic of what I will be showing next, which can actually absorb light and thereby frees up some electrons that are used in a cascade of processes to carry these electrons all the way to this ATP synthase complex, where then ADP is converted to ATP. And this is kind of the chemical fuel that every cell needs to perform its functions. During this whole cascade of events, also protons are being pumped, and these again are used as an energy source. So a key player in this whole process is this kind of small molecule, a plastoquinone, which actually is able to pick up this electron together with a proton from the Photosystem II complex, and then transfers it to the next enzyme in this whole process. And so we are particularly interested in the process of this binding and unbinding of this plastoquinone molecule to the Photosystem II complex. So we started out actually performing simulations of the Photosystem II, which is a beast of a complex, as you can see from this image. It is actually a dimer, and each monomer consists of 20 individual protein subunits. It also hosts a lot of cofactors that are actually needed to kind of trigger the energy of the light that is being captured all the way to the reaction center, where actually this electron is being freed. This whole complex is sitting actually in a special lipid membrane that has these funky lipids, galactolipids. So we also had to build and validate models for those. But eventually, we managed to simulate this large protein complex in this realistic membrane. And then when my student actually first presented this movie to me, I was quite disappointed because, yeah, we put a lot of effort in constructing these models. And then when you see this movie, it's actually very boring. I mean, you see the lipids, yeah, the gray beads are just the heads of the lipids. So you see the membrane kind of undulating around the protein, but the protein itself seems to be a very rock solid piece. But yeah, when you think about it, its core role is actually to capture the light and funnel it to the reaction center. So you should probably not expect large conformational changes or anything other interesting happening. And then if you actually zoom in to a part of this protein, then you see the real interesting stuff happening. So here we look at a part of the protein where there actually is a so-called plastoquinone-plastoquinol exchange cavity. So this is this plastoquinone molecule that I was talking about that will, in this process, be reduced from plastoquinone to plastoquinol, picking up electrons and protons. And that happens especially, particularly at this place. So this was already known because the crystal structure of this whole complex has been resolved in the presence of this cofactor. So it's nicely bound here. And this is the place where the chemical reaction eventually occurs. But what is not clear is actually how this molecule can first bind there, how it can enter this cavity, and then also how it would unbind. So based on the crystal structure, it was hypothesized that there might be two places where this cofactor could enter and leave the channel. But the function of these channels was not really known, whether one was only used for the entrance and the other for the exit. There were all sorts of hypotheses put forward. But we looked then at our simulations. And then actually we did a couple of different types of simulations. So first, we just placed the plastoquinone at this binding site, hoping that it would nicely stay bound at that position, because that's what you would expect to happen. As long as it does not get reduced, it should be binding to this binding site. And this is indeed what happened. So we did simulations showing that this plastoquinone nicely stays bound at this particular spot. But then the interesting stuff is when you reduce this molecule and make it into plastoquinol, so then we change the topology. And then we found actually that the plastoquinol is not stably bound, but actually leaves this cavity. And this is depicted in this movie here. You see it wriggles itself out of this cavity, indeed using one of these hypothesized channels, channel number 1 in this case. Now, this is only one event, but coarse grading allows you to do many of these kind of simulations. So we could gather statistics on many of these unbinding pathways. And then we actually discovered that both channel 1 and 2, with about equal probability, were being used as an escape. And even a third channel was found in this way that was also being used. So that was already a very nice finding. And then we could also study actually the reverse by having plastoquinol now in the outside thylakoid membrane and seeing it spontaneously binding to the binding site, again using these 3 channels. To make a long story short, I just show you this, this movie, which kind of summarizes now our understanding of how this exchange process is happening. So here you see some of these cofactors sitting in the membrane, and we're going to follow one in particular, and we're going to now see the dynamics. So in the central part, you see this Photosystem II dimer, and you quickly see that this plastoquinol is binding at the site of the membrane, or at the site of the membrane protein, which is close to the entrance of this cavity. Here again are these 3 channels that we have discovered through our simulations, and if the simulation continues, you see it now entering this cavity through one of these channels, which is happening right now here, and then it actually spontaneously binds to this binding site, where then a chemical reaction should take place. Well, in our simulations, we don't do any quantum chemistry, so here we just change the topology into the reduced form, and then we will see that it actually will unbind, as I have already shown you also before. Okay, that's for the plastoquinone exchange of the Photosystem II. Now a completely different kind of application, where we actually look at gene transfer. So genes can actually be delivered to cells. You can wrap them inside a lipid nanoparticle, and one of these is called a lipoplex. And these are being endocytosed. They are taken up by the cell, and then these nanoparticles, they end up in an endosome. And actually there they are being trapped, and if they don't escape from the endosome, eventually they will be degraded, and then you lose your cargo. Then if you transfer genes this way, then your genes are lost. But if it can escape from the endosome, it can actually diffuse into the cytosol and eventually reach the nucleus and being transcribed. And this is what you want to do if you want to do gene delivery. This is also the thing that is used in the corona vaccines that have made our life so much easier. In that case, the mRNA is being delivered in a very similar way. But we were simulating at the time these lipoplexes, which are mixes of double-stranded DNA, actually, which is depicted here in yellow, with a coat of lipids. They are containing some special lipids, DOTAP lipids, that are cationic charged. So the cationic head group of this lipid can nicely bind to the negatively charged of the DNA, forming this kind of inverted hexagonal structures. So assuming that these particles have been endocytosed and now need to escape from the endosomal membrane, we wanted to shed light on the actually escape process, because that is clearly something that is very difficult to study experimentally, because that happens on a very small scale inside the cell. But with simulations, of course, we can in principle look at this in molecular detail. And this is the type of simulations and the information you get out. So here you see an initial placement of this lipid nanoparticle, this lipoplex, close to a model of an endosomal membrane. And then you see there's a couple of stages which eventually lead you to complete fusion. So you see a spontaneous fusion of this particle, this lipid nanoparticle, to the endosomal membrane, allowing eventually the genetic material, these strands of double-stranded DNA, to be transfected across the membrane. And this little movie shows you also the dynamics of this process. So here the head groups of the lipids are just displayed as little sticks. And you see here that there's already a connection between the endosomal membrane and the lipoplex. And eventually a first fusion hole is opening up, allows the passage of the first strand. And then you get some kind of a complete rearrangement of the lipoplex particle, which allows you, or which allows also for the other strands to escape. So this at least gives you some idea of how this fusion of these particles with the endosomal membrane happens in detail. Now you can also do that. You can do this high throughput, so you can study this many, many times. And then you can, for instance, start looking at what is the effect of different type of lipids in this lipid particle on the overall escape probability. And this is plotted here. So here you see a survival rate as a function of simulation time. And the faster this decays, the faster the fusion actually happens. So in this case, we show that if you have replace the DOTAP lipids with lipids that have the same head, but with tails that are more unsaturated, polyunsaturated, actually the fusion happens even much more efficiently. And this is something that we also have encountered in other assays, that fusion typically proceeds faster if you add these polyunsaturated fatty acids, the PUFAs. So this kind of information can be used then by experimental labs to optimize these formulations to make this escape process more and more efficient, to avoid losses of your genetic material that are being degraded. All right. Oh yeah, then I have this slide because we also are able to scale this up. The examples I showed you were for particle sizes that are actually smaller than that are used experimentally, because then we can really do many of these type of simulations. But of course, we also want to study the size effects. So we also studied much bigger systems where we really embed one of these realistically sized nanoparticles inside a small but complete endosome model. And also there we could study how the escape of the genetic material proceeded. And if you're interested in that, you can look at this paper that we published a couple of years ago. All right, the final example, and something that is very recent that I want to share with you, is the modeling of entire cells. So yeah, entire cells are of course quite big from a simulation standpoint, especially if you want to include all the molecules. So we selected a very small cell, and there is this a so-called minimal cell that has been developed in the Craig Venter Institute. That is kind of the simplest cell type that we know of, and that is still considered to be alive. It can divide itself, go through multiple life cycles. So it is a living cell, but stripped down from all the non-essential genes. So that is nice, of course, that it is that it's small and that it is also fully resolved. So that means that we can actually start doing simulations. So it has a single chromosome with about slightly less than 500 genes, coding for 450 different kind of protein types. In total, there's about 77,000 individual proteins inside the cell. Hundreds of ribosomes are present, millions of metabolites, and also millions of lipids that actually form this surrounding envelope, keeping the whole cell together. So it is a complex beast of a thing to simulate, but at least it is that we know what should be in there to provide a realistic starting state, at least. So we set out to build an initial model of this cell using our Martini framework. So this is kind of an integrative modeling scheme that we use where we rely on experimental data like cryo-ET for the cell shape and for the position of the ribosomes, cryo-EM to resolving all the complexes that are inside, a lot of omics data to know exactly how many copies of all the type of molecules should be in there. We also rely on mesoscale type of simulations that are performed by our collaborators, the group of Zen Schulten in Illinois, that are doing this at a much coarser scale and using kinetic network models to kind of go through different stages of the cell cycle. They probe the kind of reactions that are take place and how that changes the overall content of the cell over time. So we can use that kind of information also to build our starting models. We also use a lot of tools from that we developed in conjunction with our Martini model, like polyply, Martinize, TS2GG. If you're interested in these kind of tools, you can look at this paper and see what they do. But we use them to build this initial model of an entire minimal cell. Just a few or one or two slides on the way we proceeded. So we started actually by putting ribosomes. So the ribosomes, the position of the ribosomes, they're more or less uniformly placed across the entire cell, as was shown by cryo-electron tomography. And based on these positions, we then kind of started fitting in a self-avoiding random walk polymer within the spherical constraints of the overall cell shape, and also making sure that the final end of the genome matches again the beginning, because it's a cyclic genome that this cell type has. That we do at the supra coarse-grained level, and then we did some fitting of this genome to a spline, which we can then use to trace back our Martini level DNA model. And using that procedure, we eventually end up with this nice bowl of spaghetti at the Martini level, which has the ribosomes and the genetic material inside. So when I first saw actually this image, I was getting a little bit worried, because it looks already like the whole cell space is fully occupied with the ribosomes and the genetic material. And we still had to place all the proteins and metabolites. And so I was thinking, is this gonna work, or did we maybe miscalculate? But if you zoom in to the actual model, then you will see that there's actually still a lot of room in between of these spaghetti and meatballs for the sauce to be placed. And if you zoom in further, then you see the individual bases of at the Martini resolution. All the individual bases are being represented, but still also in a lot of empty space where we can then place the rest of the cytosol. Now, this is then the final model that we made, which you see here, the envelope, containing the lipids and the membrane proteins that we put in. And inside you now see really that we filled up also the cell with all the cytosolic proteins in blue. The metabolites are probably even too small to be visible here, but also all the metabolites are in here. So this is the first cell, I think, that you will see at such a high level of resolution. But now, of course, the challenge is actually simulating this large system, which turned out not to be so easy. Oh yeah, this is a slide that shows you that indeed we packed about 70,000 proteins in here. 500 ribosomes are all there, the 500 kilobase per genome, and the cell membrane, plus almost 2 million metabolites, for a total particle count of about 600 million, when we also put it in a solvent layer in our periodic simulation box. But as I said, simulating it was actually not so easy, and still is a challenge. So originally we could not even minimize this system, because the MD software that we're using, GROMAX, was not capable of handling these large-scale systems. Fortunately, we have been working fruitfully together with the GROMAX developers in resolving these issues, and we're now at a point where we can start simulating. So we're planning to run simulations on some of the largest supercomputers that we can lay our hands on, and then we hope to get tens to hundreds of microsecond dynamics out of this system. And then some of the key questions that we would like to answer is to get an overall understanding of how homogeneously everything is being distributed in this cell. Is everything really randomly mixed? I don't expect this, but it would be nice to see which type of proteins cluster maybe together, whether there is depletion or enrichment layers close to the membrane envelope. In general, how often do certain proteins meet each other, or proteins and metabolites? Do the metabolites maybe bind to specific places in the cell that we did not foresee? Many of these questions also about the dynamics, whether a lot of hindered diffusion is taking place, or whether there might be places where there's dynamic corridors where things can move much faster. So these kind of things we hope to get out of the simulation that we are planning to run over the next year, ideally. We have already started running some simulation on some smaller scale systems. So here you see a simulation of just 1/10 of the full chromosome, just to see if our chromosome model is reasonable. And from this kind of simulation, you can, for instance, calculate a persistence length. So you can plot the correlation factor over the length of this polymer and study the decay or measure the decay. And then you expect a kind of an exponential decay to which you can fit an exponential and get a persistence length. When we do that, however, you see a kind of oscillatory decay. And this is because you have this confinement. So you can correct for that, or you can take this into account. This gives rise to a small additional cosine factor, as shown here. And then you can fit this entire equation and still get a persistence length out of this that actually nicely matches the experimentally known persistent length of double-stranded DNA. So that's one way we study smaller scale systems. And we also look at the cytoplasm, because the cytoplasm itself, such a highly crowded environment, is of course very interesting. So we've started running also 1/10 of the full scale. So we scale down the volume by a factor of 10 to start running these kind of crowded cytoplasmic solutions. And you already see, this is only a 100 nanosecond simulation, that there's an interesting kind of glassy type of dynamics occurring. You can zoom in, and then we come back to this movie that I showed at the very beginning, where you see the proteins diffusing around, starting forming complexes. Here you can also see the small red molecules, all these metabolites that are start binding to various proteins, but also to some of the double-stranded DNA. And of course, this will be a hell of a job just to analyze all the potential interactions that we are observing here, and then derive some statistics from that. But that is what the point where we are at the moment. So with that, a prospect on even what is beyond that. So we want to couple the kind of simulations at the Martini level also to this lattice microbe simulations of our collaborators that are probing this reaction and network dynamics on timescales that cover the entire lifetime of the cell. And then we can do forward and backward mapping, and information from the Martini simulation can be used to improve these network models, and vice versa. From the network models, we can get more up-to-date compositions at different stages along the cell cycle. And then eventually, of course, we hope to be able to extend this approach also to model other bacteria, E. coli, or eventually even a yeast cell, but this will be really many years from now. Okay, with that, I think I should start thanking everybody. I have, of course, many people in my group, and also former collaborators or former group members that are still continuing this Martini development. I have my current group with many talented people. Jan Stevens, I haven't pointed out yet. He is the one that is actually working mostly on this whole cell models. Bart Bruinings is the one that is on the lipoplex simulations. And then these collaborators on the whole cell at the University of Illinois. And with that, I thank you very much for your presence, and I'm here to ask any type of question you might have.